My name is Tori, and I study neuroscience, philosophy, and psychology in the College of Arts and Sciences. So, my project is entitled Using Prairie Voles to Model the Effects of Social Relationships on a Cell Called Microglia. So, what are these prairie voles, and why do we use them as a model species? Well, first of all, they're a non-commercial available animal model, and so that's why many of you probably haven't heard of them before. They look pretty similar to mice. Like mice and rats are probably the typical animals you think of for lab experiments. But the reason we're interested in them behaviorally is because they exhibit something called social monogamy and selective pair bonding, which I'm going to introduce on the next slide. Finally, we're interested in them because they show really robust sex differences between the species. So we see really dimorphic differences in various biological phenomena between males and females. So what is this pair bond that I mentioned? Well, in animals, we define this as a lifelong bond with biological correlates that forms between two mating partners. And this leads to various biological changes in hormones such as oxytocin and vasopressin, and also in hormones such as dopamine, which are associated with reward learning. So there's a lot of research as to whether this pair bonding behavior is perhaps a reward-seeking behavior in these animals. So what does this pair bonding allow us to model in humans? First, it allows us to model romantic and/or sexual partnerships, perhaps most directly analogous to what you would think of in an animal. But additionally, it allows us to model long-term friendships and selective caregiver attachment that forms when you're an infant. So my project is about the relationship between prairie voles and this cell called microglia. So what are microglia? Most of you, when you think of brain cells, probably think of neurons, but we actually have other cell types in our brain called glia, which are non-neuronal, and microglia are a type of them. They serve many functions, one of which is surveillance of the brain tissue. A second one is phagocytosis, which essentially means the cell cleans up debris by sort of engulfing it. And they serve all of these other functions that you can see on this diagram, while very important to the functioning of a healthy organism, not super essential to my project. So the two I want to highlight are synapse monitoring and pruning, and inflammation. So when we talk about a synapse in the context of my project, that means the connection between two neurons. When we talk about monitoring and pruning, we talk about cells who have the ability to change the number of connections between neurons, change the strength of connections between neurons. That's a phenomenon we refer to as plasticity, and that's how learning and memory occurs in the brain. So these microglial cells play a really critical role in learning and memory. Additionally, they play a really important role in the inflammatory response of the brain. So one of the ways that we've studied microglia throughout history is by classifying their morphology. So this diagram is an example of a research paper that classified microglia morphology into 5 distinct subtypes. However, the two my project focuses on is the spectrum of homeostasis to reactivity. If you're not familiar with the term homeostasis, it essentially refers to the optimal functioning of an organism. So at peak health, however that organism is functioning is referred to as homeostatic, and that's the morphology associated with this homeostatic phenotype. You can see on this spectrum that homeostatic microglia are associated with a smaller cell body and more processes, whereas reactive microglia are associated with a larger cell body and fewer processes. So before I give you my research questions for this project, it's really important that I clearly define this intersection between prairie voles and microglia. So as I mentioned, prairie voles exhibit this pair bonding period, and this pair bonding period is a period of plasticity in the brain. Additionally, there are species that allow us to model human social behavior and those 3 types of relationships I mentioned at the beginning. Microglia are a cell type that are a key component of this synaptic plasticity. Additionally, they're an indicator of inflammation in the brain. So where does the intersection lie? The intersection lies in this potential role for microglia in inflammatory hypotheses of disorders of human social behavior. Very quickly, I'm going to outline how immune hypotheses of stress disorders came to be and how this allows us to model social behavior. So, when we see symptoms such as social isolation in disorders of social behavior, that increases your body's release of the stress hormone cortisol. And one of the effects of having circulating amounts of cortisol in your body is increased inflammation. Then, when we connect these disorders to pair bonding, disorders, such as major depressive disorder, are characterized by these deficits in social relationships. And using a model that exhibits pair bonding allows us to experimentally assign the presence or absence of the significant social relationship in the experiment. So we can isolate the social relationship variable to look for the effects of whether or not a social relationship is beneficial or detrimental. So what were my research questions in this project? First of all, I was interested in how microglia in pair-bonded prairie voles compared to the microglia in unpaired voles. Second, I was interested in whether the sexual dimorphism of microglia morphology that had been previously established in other animal models persisted in pair-bonded versus unpaired voles. Finally, I wanted to know what the results can teach us about the importance of using socially monogamous species to model human behavior. My methods involved, very simply, staining microglia. This began with sectioning brain tissue, staining it, and imaging it. And this is an example of what the slides look like. My imaging results were able to provide characteristic examples of homeostatic and reactive microglia in the brain. On the left, you can see a homeostatic microglia with a smaller soma and extensive branching. And on the right, you can see a reactive microglia with a larger soma and reduced branching. Additionally, I was able to create a montage of photomicrographs showing that as soma size increases, branching decreases. Finally, I was able to analyze the results of the cell body size, also called the soma, of the microglia. First, I found no differences between the brain regions that I studied. Second, I found that all groups were significantly different, except for paired males and paired females. Third, I found that the largest difference existed in somal size between unpaired males and unpaired females. And finally, I found that there were no significant differences between unpaired and paired voles unless you separated them by sex. The important conclusions I was able to gain from my data is that the pathway from unpaired microglia to paired microglia is sex-specific. Female microglia showed increased reactivity after pair bonding, whereas male microglia showed decreased reactivity after pair bonding. Finally, I want to highlight the novel objectives that my study achieved. First, I validated a new type of protocol in prairie voles. Second, I was the first study to examine microglia in the vole anterior cingulate and prefrontal cortices, which are my brain regions of interest. And finally, I was able to provide evidence for the persistence of sexual dimorphism of microglia across unpaired and paired groups. I'd like to end by thanking all the people that have supported this research project, and particularly the Europe Global Challenges Research Award, which funded this project for an entire year. Thank you very much.
Raise your hand and I'll bring the microphone to you. And also, please make sure to use the microphone so those on Zoom can also know what your question is. Thanks.
Hi, I'm curious about some of the previous research that you've looked at in terms of sexual dimorphism, specifically with the prairie voles and their neurological morphology.
Yeah, absolutely. So this is a condensed version of a 45-minute thesis defense in the Department of Neuroscience. So I know a lot. Essentially, prairie voles are really understudied because they're not commercially available. What has been previously found is that there is this sort of sexually dimorphic baseline where in many circumstances, whether it's you're looking at microglia as a cell type, you're looking at hormone levels, at baseline, so unpaired species, they'll be really sexually dimorphic, and then when they're paired, they look more similar. And if you separate them, they return to that sexually dimorphic baseline like after separation. So it's very dynamic based on whether they're like in the presence of their partner.
Yeah, thank you for your presentation. And I was wondering if you could talk just a little bit more about why specifically you chose to focus on anterior cingulate cortex and PFC.
Yeah, for sure. So that's actually one of the biggest objectives of this study is that most of the prairie vole research is done in reward learning areas, such as the nucleus accumbens, the periventricular nucleus of the hypothalamus. And that's because of its associations with dopamine, which are very valid, but I'm more interested in modeling things like major depressive disorder and disorders of social functioning, which in humans, the areas that we study are the anterior cingulate and the prefrontal cortex, because those areas have more to do with sort of like your social value and how you sort of value your social relationships. And so pivoting to those brain areas is something that hasn't been done before.
Yeah, I was just looking at the abstract about the major depressive disorder that you begin with, and I'm curious if any of these implications you've connected to that aspect of the research, and what how for somebody unaccustomed to the field we can connect these ideas.
Yeah, definitely. I wish there was more time to talk about it, but essentially, one of the motivations behind this research is that the current pharmacological treatments available for mood disorders, so that's major depression, anxiety, OCD, etc. You see really sex-specific effects of the pharmacology. So females tend to respond better to SSRIs. Males tend to respond better to an older type of drug called tricyclics. And so since we see those like sex-specific effects of treatment, we know that there's something sex-specific going on with the pathology. And so there are several models that also show when you get rid of the pair bonding aspect, when you get rid of this like social relationship variable, and you just look at organisms which have been, they're either non-psychiatric, and then you stress them out and you create some sort of psychiatric phenotype, you also see the sexual dimorphism of they start at a sexually dimorphic baseline and they converge onto some sort of reactive phenotype in stress. And so that sort of connects to all of this, adding the social relationship variable in, but is motivated by trying to improve pharmacology.